Well, I'm very pleased to introduce our final panelist. This is Dr. Ralph Zitnik. Dr. Zitnik is a pulmonologist who trained and did research on faculty at Yale. He went from there into industry almost 18 years ago. And he's done research, biotech clinical development, and in the inflammation therapeutic area ever since, working at Pfizer, Immunex, Amgen, on a number of drugs, including Enbrel, a TNF antagonist, and Prolia, a rankle inhibitor. Dr. Zitnik has been at SetPoint Medical a little over 5 years now, and SetPoint has just completed the first ever study of an implantable medical device for rheumatoid arthritis, RA. Welcome, Dr. Zitnik.
Okay, great, thank you. So, again, my name is Ralph Zitnik. I'm from SetPoint Medical, and you've heard today from three excellent speakers that have told you the aspirations, where we're going, what the future holds, et cetera. And right now you're going to get a report from a platoon sergeant on the front line, because we're out there doing this today, and I'd like to share with you kind of what we've done over the last 5 years. And for those of you who are investors or developers or thinking about going into this area, just to let you know sort of what we've experienced over the last 5 years as we've tried to move towards these aspirations that the other speakers have outlined. Not advancing here. Well, we're based in Valencia, and I'll just read you our mission statement here. We're developing a unique implantable therapy to improve the lives of patients with debilitating inflammatory diseases. And as you heard from Victor, one of the really most important things about doing this with medical devices is you can cut costs. And so our goal is to reduce the treatment costs of these expensive inflammatory diseases by half. You can see our investors. We have Action Potential GSK, Boston Scientific, Covidian, and three venture firms, Foundation Medical Partners, Morgan Thaler, and Topspin. So developing a bioelectronic medicine. You've heard a lot about this today. It's a unique opportunity. It's a huge market space, as Victor showed you. It has huge opportunities, but not without some accordingly unique challenges. And I guess, as I said before, what I'd like to do today is show you how SetPoint has faced these challenges and what we've learned that can potentially help you as either a developer or an investor. And I'd like to focus on preclinical development. What did we do? How did we face this? The translational medicine issues, as Jeff talked about, these are really important. And then what will we face, and all of us face, as we move on to later stage development and large controlled multi-center studies. So we've talked today a lot about bioelectronic medicines, and here's again this list of aspirational characteristics. We've talked a lot about information resolution at the individual neuron level, that the device has to read, it has to interpret, it has to react and respond. And these are very important things that we need to do that we're not there yet right at this moment. We talk about using peripheral nerves to affect visceral organ function, and we've seen that a couple of times this afternoon. And I think really most importantly, the key thing is that these devices will treat prevalent and chronic diseases that are traditionally served by pharma products and biotech products. We're talking about atherosclerosis, hypertension, diabetes, big diseases that are traditionally treated with pills and biologic agents. Well, I think the top two bullets really represent the future. Where are we going with a lot of engineering and science work? Where will we get eventually? This is the place where we need to be in the future. But I think really today we're there in certain ways. And as I'll show you here, we have used a device on a peripheral nerve to affect visceral organ function and treat a systemic disease, specifically one disease that I'll show you about in autoimmunity. Here's the mechanism, and as Juan Pablo said, one of the key things about this new approach is we have to be very mechanistic. We have to understand how and why it is that the charge that we apply to a nerve really affects the organ system that we're studying. And on the left-hand panel here, I'll show you the cholinergic anti-inflammatory pathway. It's a neuroimmune pathway that was really described about 20 years ago by the founder of our company, Dr. Kevin Tracy. And this pathway is very detailed and very involved, and that slide on the left there summarizes about 15 years worth of work and probably 200 papers. But it's a reflex that basically the body uses to modulate the immune and inflammatory response to tissue injury. And just like blood pressure or heart rate, it keeps the amount of inflammation and immunity appropriate to the level of tissue injury. And inflammation is sensed, specifically TLR-driven inflammation is sensed in the peripheral nerve and in the brainstem. There's an outflow of activity through the vagus nerve, through the celiac ganglion to the spleen. And I'll draw your attention, those of you up front, to that little bubble up at the top. And there's a key cell there called a macrophage, and a key receptor called the alpha-7 N-acetylcholine receptor. And this cell really responds to a neurotransmitter, acetylcholine, and we can demonstrate this both in vitro and in animal studies, that when that cell in the spleen or passing through the spleen sees acetylcholine, it becomes unable to release TNF to the same extent for a period of about 24 to 48 hours. So what we get in both the spleen and the intestine is the ability to essentially quiet the release of these cytokines in organs that are abnormal in diseases like rheumatoid arthritis and Crohn's. And on the right-hand panel, you can see our therapeutic approach to use a small, as we've heard today, miniaturization is important, a small implantable device that a patient can have implanted and could potentially use instead of a biologic agent that's quite expensive and has substantial safety risks. So this is our overall approach, and let me walk you through how we got to this approach in clinic. And we've talked again today about the differences between what you do when you do the preclinical development of a drug or a biologic agent versus an active implantable device. With a drug, of course, you have a dose. You measure the effect of that dose through a pharmacokinetic response. Typically, as many have pointed out before me, there's a biochemical reaction at a receptor that you measure as part of a biomarker, and then that translates to an effect on a disease model and a clinical study. This may be an oversimplification, but with an implantable device, you're stimulating a nerve, the muscle, or the conduction system. You're looking at a physical effect to some extent. You look at the stimulation settings, as we just heard. Those are very important. You read out the effect of those stimulation settings in an ECG, an evoked potential, a heart rate, etc. Your measurement really is physiological, but in the end, your effect is on the disease model, and ultimately you hope on the clinical study and the disease that you're treating in people. Well, I guess I'd put it to this group that a bioelectronic medicine as a preclinical developer will require a hybrid approach. Certainly if you're modulating the immune system with an electrical input, you can't either do the left or the right-hand panel. You have to do a bit of both. So when we started out, at least, to do this 4 or 5 years ago, the real questions to us were, how can we determine the optimal stimulation paradigm? As Jeff has said, that's really critically important and can make or break both a preclinical study or a clinical study. The models for the diseases that we use in immunology and inflammation are almost always in rodents, and oftentimes in mice, genetically modified mice. So one real question for your preclinical program is, can you build hardware that fits into a mouse? That's not an insubstantial problem. And finally, of course, does that hardware, especially in a small animal, affect the readout of the model in a way that can confound you? So as we thought about this 5 years ago, here's the approach that we and our collaborators took. So the traditional pharma rheumatoid arthritis model is called collagen-induced arthritis, and it's a way of developing an analog of human rheumatoid arthritis in mice and rats. And the approach we took really was a combined approach, sort of the classic genetic and biochemical approach combined with a typical medical device approach. So we and others gave nicotine or an agonist, a pharmaceutical agonist, of this alpha-7 N-acetylcholine receptor to our animals and found that the classic pharma model that one always uses, the animals got better. As you'd expect, when we cut the vagus nerve, the animals got worse. When we went in and did a genetic knockout of the alpha-7 acetylcholine receptor, as one would expect, the animals that lost that critical receptor got worse as opposed to the matched litter mates. Another group that we don't collaborate with did a really interesting experiment where they sutured the vagus nerve up against the sternocleidomastoid in the neck of a rodent and got essentially a poor man's vagus nerve stimulation device. And they indeed also found that the disease improved. So we came along then and said, well, let's use a standard cuff electrode like one uses typically in medical device work. And you can see this here. It was designed by Yakov Levine, one of our engineers. And You can see it being put around the vagus nerve on the front of the mouse, and you can see the little guy with arthritis down in the bottom right-hand corner here getting his nerve stimulation treatments every day. And we were able to work out a set of stimulation paradigms that gave us this result, which we just published a few months ago, where the clinical signs and symptoms of arthritis up at the top are reduced by about 50%. But more importantly, we were able to reduce histological inflammation, pannus and destruction of the joint itself. And so the things that really make a rheumatoid arthritis patient debilitated eventually in our studies and in all of these studies were made much better. Well, we then moved on to this, from this to our first study. We took these 4 studies as evidence that we should try this in people, and we did the study that you can see here. And it's an open label, single-arm study, pilot study, where we took patients and implanted them, gave them an escalation in their stimulation over the course of about 4 weeks, went from the once daily stimulation that we typically do to 4 times a day if they didn't respond by 4 weeks, stopped, started, and then treated again for 3 months. And the endpoints of the study here are very typical ones for rheumatoid arthritis studies, the ACR, American College of Rheumatology response, and the DAS score. We did this to start with a commercially purchased Cybronic system with a couple of modifications to the way we deliver the stimulation. We did 2 cohorts of patients. The first cohort of 8 patients were biologic agent naive patients. These were patients who had failed methotrexate, the standard first oral drug for RA, who would otherwise have been a candidate for a TNF antagonist. We wanted to essentially benchmark this population against drugs like Humira, Enbrel, et cetera. Then when we had good results in that population, we came back and said, well, what about a more typical medical device type population, patients who had failed multiple biologic agents, not just one, but at least 2, and not just one mechanism of action, but at least 2 separate ones. So I'll show you the results from the first cohort here. These are the first 8 patients, and on the vertical axis is the DAS score. A higher score means more joints, more inflammation. A level of 7 or 8 would be a patient that was really chair-bound. A level of 2 to 3 is a patient that has good disease control, and as you can see, a level of about 2.1 or 2.2 constitutes disease remission. And what you can see is 6 of our 8 patients had really wonderful responses, and 3 or 4 of them went down to the level where they were essentially remitted. And 2 patients, for reasons that aren't entirely clear even now, didn't have a response. So we also saw in these patients that objective measures of laboratory inflammation like CRP and other mediators improved, so that it wasn't just subjective responses like joint scores. The markers and mediators that we measured during the study got better as well. And I can't show you the data yet on the cohort of 10 late-stage patients because we haven't presented it at the rheumatology meetings yet, but I can just tell you verbally that a little over half of the patients had responses that were in this range. Some of these patients had failed 4, 5, 6 drugs, so they were really patients who you would expect not to have a big placebo response, and for whom there really aren't a lot of other potential treatments. So in both early stage and late stage RA, in a total of 18 patients so far, we had some really interesting results. Let me just quickly tell you what we learned as we did this study. And again, you're talking about doing studies with medical devices with internal medicine experts who are not used to doing medical device studies. So from the beginning, they're skeptical about using medical devices. You need education, good science, data-driven arguments. Internal medicine subspecialists, as many of you know, tend to be surgery averse. So rheumatologists, endocrinologists don't like to send their patients to the operating room to have hardware put in. So one of the things we found in doing the study is you have to go as the sponsor of the study and find the surgeon and find the internist and bring them together, especially when it's a neurosurgeon and a rheumatologist. One of the other important things we realized is that you really have to use surgeons very effectively as persuaders to talk to the internal medicine subspecialists. Tell them, this is not so hard to do. We put medical devices in every day. We'll work with you on this study, and we'll make it work for you. Similarly, the staff at their sites, the clinic nurses, the research nurses in an endocrinology office, unlike a cardiology office, don't feel comfortable programming devices. You really have to be there to help them on this. These trials are harder for them to do than sort of a classic pill study or a biotech study. And competition to get patients into a study is always fierce. So you really have to be there for them and with them to help them learn and feel comfortable with devices, surgery, device implantation, and programming. And finally, one of the things we learned as part of doing this study is that even if the physicians, especially the internists, are a bit hesitant and a bit skeptical, patients will drive this. And so many of the speakers today have talked about these diseases, the number of patients that have these diseases, and the need for something new besides just another drug or another monoclonal antibody. And what we found out in Amsterdam, which was one of our main centers, was when our principal investigator went on the news and showed a picture of, like you've seen today, of one of these small, slick devices and so forth, his center got 2,000 calls. And so the pent-up patient demand for these types of alternatives to standard drugs and biotech products will help you drive recruitment and success in clinical trials. So here's our device. As I said, we did the first study with Cyberonics. Here's our platform, and we talked today about the importance of miniaturization. It's a self-contained leadless cuff you can see up in the upper left-hand corner. It fits on the nerve. You can see the two electrodes there, and it's held onto the nerve with this thing we call a pod. Looks sort of like a hot dog bun. Holds the nerve in place against the electrodes and ensures really robust activation of the nerve. The patient wears a collar for charging and telemetry just for a minute or two a day in order to charge the batteries, and when they're in clinic for the physician to change the settings. And it communicates this collar with a standard iPad app via Bluetooth. So we're now going into our final pre-GLP study next week. We hope to have this system into the GLP first-in-human enabling study during this year and to implant the first of these devices, likely in a rheumatoid arthritis patient before the end of 2015. So what about late-stage considerations? Now we're ready to move into later-stage trials, bigger studies, multi-center studies, et cetera. Our first study likely will be in this multiple biologic RA patient population, but we're also starting a Crohn's study this week. And so if the Crohn's study is positive, we may also or alternatively do Crohn's. So there's challenges and opportunities with this late-stage piece, just like with any development program. One of the things I think you'll have to realize, and Victor spoke to the fact that these development programs can be less expensive, but I'd caution there that the communities in endocrinology, in rheumatology, in cancer and so forth are used to big studies. Bigger programs with more patients in them than you may be used to doing. The other thing we talked a bit about is that the controls. Again, the standard for a pharma study is a placebo-controlled RCT. And a lot has been written, I know many of you in the device community are familiar with this, about the difficulties and challenges in sham and blinding studies with medical devices. So it's something to think about as you move into these studies. You have to use typically the same efficacy and safety endpoints that are used. So if you're doing a diabetes study, I think it's highly likely that you're going to have to look at hemoglobin A1C. When we did our RA study, we weren't able to make up a new endpoint. We had to show that this worked on the same endpoints that the medical community was used to seeing. I think you'll probably face that same challenge. And then finally, comparative health economic considerations. So Victor talked about this a bit today. Our device will have the cost of the device and the cost of the implantation, which are not insubstantial. But typically these biologic agents that are used for RA cost anywhere from $15,000 to $40,000 per year. So the healthcare system really starts to break even and turn positive on a medical device after about a year or two in this patient population. So I think we've really, with this device, while it doesn't read, react, and respond, it's a small, miniaturized, very complex device that's gone into a high-cost, severe, high medical need, systemic disease, and holds great promise to be a new kind of treatment. So there is a big opportunity, there's a big medical need, and as I said before, as we learned in Amsterdam, patient demand will drive these studies and will drive adoption. So in summary, bioelectronic medicine is more than a concept, it's more than a drawing board issue. We're, at SetPoint at least, and other companies may as well, be doing this today. We hope that relating the experience that we've had over the last couple of years will be helpful to you as developers or investors. I think our study indicates that in RA and Crohn's and other similar diseases, this approach might be a great alternative for patients in the future. At every stage in development, there are challenges, just like with any sort of product development. And I think one of the things you have to remember if you do these kinds of programs is you can't think just like a drug developer or just like a medical device developer. You have to do both, and you have to take a hybrid approach. And I hope, as you've been convinced by all the speakers today, that this is a really unique and exciting field that's just in its nascence. So thank you for your attention.
Questions?
Yeah, this is a great presentation, really exciting work. Regarding the study and the 8 patients, what was the adverse event profile that you saw? I mean, I'm sort of from the drug world, and I'm not even used to thinking about what sorts of adverse events you might see with a device implantation study.
Yeah, it's a great question. We didn't see opportunistic infections. We saw the typical kinds of one adverse event in a patient who had the implantation, who had hoarseness for a short period afterwards. This is well described when you operate on the vagus nerve because of the recurrent laryngeal. Nothing really unusual. Now, we've got the latest, the longest patient in the follow-up study out 3 years, and we've seen no TB, we've seen no pneumocystis or opportunistic infections, and good disease control long-term in the majority of the patients.
This question is for Dr. Ardell. Relating to your the neurocardial procedure, how do you factor in intersecting behavior change with treatment?
I'm not sure I understand what your...
Patient behavior, you correct the, you correct.
Right.
And then how do you guide the patient in terms of behavior change to not have to come back for an additional procedure?
Well, what we actually had, there was a nice study we just had published in terms of, we actually did behavioral assessments on patients being treated with the sympathetic procedure that I showed you. And actually the patients in terms of their quality of life scores, in terms of their ability to do everyday activities, actually improved quite dramatically. As we've referred to over here, there is always some arguments about placebo effects. There's also concerns about how you implement this therapy, and some of the device approaches actually have some adverse effects with it that if you are smart about it, you can actually get around those procedures and actually get to effective therapies. But overall, the experience, at least in heart failure, at least from multiple studies, has been patients who have been essentially been quite restricted actually can start living quite better, very much more fulfilling lives, doing a lot of basic things. So, does that answer your question?
Yes and no.
Well, I like the no part.
We're talking about behavior change, not just, going back to the same routine of having four ways of living.
Right. Okay, I see what you're saying. Right now, many of the device therapies in terms of especially cardiovascular disease are at the end of the treatment ladder. Obviously, when you implement these types of procedure, you start to recover the patients. You obviously want to change their behaviors, increase exercise levels, increase diet considerations. But once again, one of the things we are hoping in the field is to move this up to when you identify patients who are at risk, getting this implemented at an earlier stage and actually help them along with behavioral modifications. Because there are inherent differences between us and there are acquired differences. You can't do much with the inherent, but the acquired you can impact, and I think that's quite important to do as well. It's all part of a full treatment package you have to do with these patients. Is that better?
That's good.
All right, thank you.
My question is, I assume that device, what is it? Oh, my name is Alok Nigam, and I'm with a company called Health Naturally. I am more with the device industry side actually, but my question is that looking at your well, this is actual picture, not a cartoon, that these devices are implanted relatively shallow. Is that a right statement?
Yes, correct.
Okay. So you will have surgical complications along with the biological response to it?
Well, this device, like most vagus nerve stimulation devices, is implanted on the nerve in the carotid sheath. It's a fairly easy surgical approach. It's been done for 20 plus years with a standard coiled lead.
So
yeah,
my next question, continuing to that, is that you will form some scar tissues and all that. Will that change the response and in the process the therapy with that?
The answer is scar tissue may, yes, change the process. Again, with the standard coiled leads, the scar tissue forms on the outside with a little bit around the nerve, and doesn't really affect the response. With our device,
Your experience has been good so far.
I'll just explain it. With our device, one of the really interesting things about it is it's a small self-contained device, and a pod, like a pea pod of fibrosis, goes around the whole device. So one of the things the surgeons have found in the dog studies is, unlike a coiled lead where you have to go in and manually dissect this corkscrew off the nerve, which often damages it, what you do with our device, if you have to take it out, is you make a quick slit, you pop a suture open, and the whole thing comes out in block. So I've talked to you about the RA piece of this, which is very interesting, but just beyond that, as a peripheral nerve stimulation device, this is a really kind of nifty, elegant implant that could be used on almost any peripheral nerve.
Oh, okay. And what about the acute surgical problems which can interfere?
With vagus nerve stimulation, I mean, if you have a surgeon who gets in and mucks around, you can damage the surrounding structures. There's postoperative infection. You know, again, these things have been studied in 80,000 patients, mainly by Cyberonics, the company that makes the epilepsy device, for at least 20 years. And I think one of the nice things about this approach, as opposed to a drug approach, one that I come from, is, you know, you can control surgical risks. You can't control, I used to work on a TNF antagonist. You don't know who's going to get multiple sclerosis. You don't know who's going to get worsening heart failure. It's a rare but catastrophic sort of side effect. Here you have side effects where, in theory at least, you can control them technically.
Thank you.
And I agree with him in terms of the vagus. We also, in terms of the experience with spinal cord stimulation, goes back about 20 years as well. And it's actually been a very good outcome with that.
Peter Blaisdell. Another question, actually. In thinking a bit ahead to the future, is it possible actually to have the patient to some extent interact with the device themselves, for example, in arthritis or whatever your disease indication is, to be able to modulate to some extent based on their response, whether, for example, pain is being reduced or other symptoms that the patient can perceive? So not only does the investigator, when they insert the device, basically have the ability to set it to have the achieved outcome from a clinical point of view, will the patient have the ability to interact with the device in a way that would, or potentially have the ability to interact with the device in a way that sort of the patient sort of sets whether they're responding the way they think they should?
So you're already thinking like a bioelectronic medicine developer. That's exactly right. I mean, it would be great to have, instead of a physician-programmed iPad app, to have something on their iPhone that they can interact with, that can read out the disease in their joints. You know, how many steps do they walk every day? Anybody that's got an iPhone 6 knows how many steps they walked yesterday. Could that be linked in eventually? Sure. I mean, that gets you to the loop that we talked about, you know, where you're reading out the disease and changing the way the device functions. But absolutely.
Great. I have a couple questions. One is for my colleague here, Juan Pablo. Given your unique position at GSK and experience with drugs and biomedicine, do you see of emergence of better drugs that could be used as adjunct with these biomedicine? Is this going to be a hybrid therapy? Where do you think we're going to go from here?
Sure. So, you know, oftentimes these are very specific to particular conditions and sort of what is best in class medical therapy already. And are there potentially, you know, arguably synergistic effects of a, you know, of a medical therapy in addition to a bioelectronic medicine? And so I think the answer from my perspective is absolutely yes. There's health economic benefit to replacing drugs in particular conditions, and certainly safety benefits, as you know, Ralph was mentioning a moment ago, in some conditions. And I think it's funny, I was on a call a couple hours ago today talking just about this issue with a company that was pitching us, and I think there are very specific examples of where, you know, should a bioelectronic intervention work well in a clinical setting, there are examples of actual synergistic mechanistic effects where you could imagine both treating a nerve and specifically some of the receptors and the neurotransmitters that are delivered from the nerve, you know, actually enhancing the therapy with a drug, both from a mechanistic perspective, but also from anatomical perspectives. I can't get into some of the details of the example I'm dying to talk about, but it's pretty exciting actually, so I do believe so.
Thank you. My next question is both for Jeff and Victor. What are your thoughts about neuromodulating the renal function to improve congestive heart failure and atrial fibrillation? I was attending a session at TCT a couple of weeks ago, and they were talking about doing procedures like that. I'd like to get your thoughts on it.
Well, the point you quite correctly make is that the renal circulation also provides the cardiovascular system with important information for regulatory function. And actually, a lot of the procedures, for example, the renal denervation procedures they're doing now, has the potential for impacting neural processing, information going to the spinal cord and brainstem, and then through that mechanism can actually have important influences on control of function. I would like to make one comment about the drugs that we're just talking about right here as well. With the electroceutical approaches we're doing, we really don't know what the neurotransmitters are in many cases that we're actually releasing. And that's important not only for understanding how the therapy is working, but it's also important because some of the drugs that you're giving therapeutically may interfere with the electroceutical approach. So we really do need to understand that, and that's one of the big opportunities available in the future too.
So I just want to, I guess, answer this question more broadly. The way that denervation works is it's anatomically removing some afferents or perhaps even efferents that activate that specific organ, or that in this case it's a kidney. What bioelectronic medicine intends to do is, instead of doing this one-time surgical procedure, instead tuning the activity of that particular nerve, which becomes on-demand kind of modulation. And actually a lot of effort by GSK, I think almost 50% of teams that are working are doing not stimulation of the nerves, but are trying to actually block the nerves, because in many cases the nerve is overactive. But it really sometimes is not helpful to completely remove the nerve. Rather, you would like to tone it down, so basically reduce it by 50 or 75% activity in the nerve, and also perhaps also reduce it at the times that's convenient. For example, if you're asleep, probably your sympathetic activity is low, so there is no point of inhibiting it. But when you're awake and active, or after dinner or lunch, maybe that's when you would like to turn on this stimulation to inhibit that particular nerve that's causing diabetes or some other disease. So to have an on-demand inhibition.
And getting back to your question about patient control, especially in angina control, actually with some of the patients with spinal cord stimulation, they can actually activate it preemptively if they're going to know they're going to go out and do an activity that's going to be potentially deleterious. So that already is there. Obviously, our long-term goal is to develop automated signal detection so we can initiate that with the device itself with patient override.
Hello? For the device entrepreneurs in the room, what are the research tools you biologists and the physicians need that you don't have yet that would be helpful to you progressing in this field? For anyone that would like to comment.
So on the biosensor side, as I mentioned in my list, so we know quite well how to stimulate small nerves. We don't have maybe very small cuffs, but we know how to miniaturize them. So that's a small technical challenge. The bigger challenge is miniaturizing some of the biosensors. Currently, as you know, many of these biosensors are big bulky machines like HPLC and others. So that will be a big challenge to getting some biochemical readouts. And some of the ways we can think of start doing that is by maybe instead of using traditional samples where you actually take a sample and then you vaporize it and do something with it. Instead, be creative, do some electrochemical approaches, amperometry or cyclic voltammetry, some techniques that are more applicable to in vivo. But as far as I know, there are very few, almost no examples right now where there is fully implantable biochemical sensor. So this will be something, a hot area of research for the next 5-10 years.
And I agree with Victor on that. And then also being able to miniaturize and get optimum neural signals from various nerves over long periods of time is obviously going to be a big opportunity as well.
I think we're running out of time, and I want to thank the panelists. Please put your hands together for them. And this is a very exciting area, and we have emerging companies here from